It is glucose 1-phosphate, the precursor. It is formed from glucose 6-phosphate by the enzyme phosphoglucomutase, right? So what is the first product? Is the end product, is the direct end product of glycogenolysis is what glucose 6-phosphate is. Is what the direct end product of glycogenolysis is glucose 6-phosphate? No. What is the end product then? Is glucose 1-phosphate that is converted into glucose 6-phosphate, and later on if the action is taking place, if the reaction is taking place specifically in liver, then glucose 6-phosphate is converted into glucose. The enzyme name is glucose 6-phosphatase. Okay, that is a very key enzyme that plays a very important role in gluconeogenesis as well as that takes place specifically in liver. Yes or no? Okay. What is the fate of glycogen degradation in muscle? ये जो terminologies है ना जो मैं use करता हूँ, what is the fate? Explain the significance of muscle glycogenolysis. This is how they ask you. This is how they are going to ask you in reasoning type questions. Justify your answer, kind of thing. How the questions are going to be asked generally. Explain the significance of muscle glycogenolysis. You don't know what is the significance. What is the significance? That is what I am asking. What is the fate of muscle? What is the fate of glycogen in muscle after undergoing glycogenolysis? Achha, it takes almost a week to get depleted. Fine. I am asking about what is the fate. Fate is if I am asking about fate, what is the significance of glycogenolysis in muscles? Then your answer should be it converted into glucose 6-phosphate, which is not degraded because muscle cells lacks glucose 6-phosphatase enzyme. As a result of that, glucose 6-phosphate directly moves into glycolysis and provides the energy needed for the muscle cells. This should be your answer. Okay? So you are well aware that glycogen synthesis and glycogenolysis are two different processes. They take place in the presence of certain enzymes. If you are talking about glycogen synthesis, the key enzyme is glycogen synthase. If we are talking about glycogenolysis, breakdown of glycogen, the key enzyme is glycogen phosphorylase. Okay, and both of them are occurring in a different phase of time. I have roughly told you that glycogen synthesis occurs when you are in well-feeded state. Your blood is flooded with glucose. You have enough glucose and you have enough energy also. Okay, in roughly I have told you. On the other part, I have told that glycogenolysis, the breakdown of glycogen, occurs when your body is in demand of glucose. That means you are in fasting state. You didn't have your food. Your blood glucose level is dropping continuously. You are going to hypoglycemia. So in the response of that stress, fine, epinephrine is released. Glucagon is released. This all glucagon and epinephrine is responsible to activate certain secondary messengers. Regarding that, it causes a covalent modification in these enzymes like glycogen synthase and glycogen phosphorylase, making them active or inactive. Okay, this was my previous statement. Now today what we are going to look around is how the presence of hormones, how the presence of glucagon, how the presence of insulin, how the presence of epinephrine, how the presence of certain allosteric effectors are going to activate or deactivate these two processes. Okay, before I jump into that, remember I have already explained you the regulation of glycogen synthesis or glycogenesis are regulated by reciprocal regulation. If I say, wherever you hear this term, reciprocal regulation, that directly means that if one process is getting activated, then the other process is going to be inactivated, and vice versa. Okay, that is what reciprocal regulation is all about. Is that clear? Am I clear? Okay. Let us start with a phase. For example, most of you didn't had your breakfast, many of you? Yes or no? Okay. Last you had your meal is at night, somewhere around 9 o'clock, 9:30. It's now 10. It's almost 10, past 10. So you are in fasting state, more than 8 hours of fasting state. राइट? क्या मैं मर गया? मैं भी रात को 10-9 बजे खाया था, सुबह उठ के देख रहा हूँ टाटा खत्म हो गया हूँ मैं. Did that happen? No. What made this thing possible? That I'm still alive. We all are alive. Because your brain cells, your RBCs, cardiac tissues, they primarily depend on glucose. Fine. They consume maximum, they produce maximum of their energy provided by catabolism of glucose. So where is that glucose coming from? You will say that, sir, gluconeogenesis is there in liver. Fine. But you know, gluconeogenesis occurs provided to, in liver, provided to that the glycogen is empty in liver. As I told you, it is only 8 hours. Is the muscle glycogen going to get depleted? No. Muscle glycogen is still there. But what about liver? Liver glycogen is, storage depleted. Khatam. Fine. So it is the first thing that happens inside the liver, that first the glycogen is utilized for the production of blood glucose, so that our glucose homeostasis is maintained. Post to that, the liver or the body depends on liver for the production of glucose provided by a process called as gluconeogenesis. Is that clear? So ultimately, these two processes are responsible for producing or providing glucose to our blood, and as a result of that, we are alive. We are living. We are taking breath. We are living. Fine. Now, how this process is regulated? What controls? How it is controlled? Whether to activate it, when to deactivate it? You have to understand this process. It's not that, it's not that critical. It's not that tough. It's a very easy to understand thing if you apply your sense a bit. Okay, just go through what I am trying to explain you out here, over here. Imagine that you didn't have your food. You are at fasting state. Imagine that we all are in fasting state. Fine. As a result of that, what is going to happen? That our blood glucose level is going to fall down. Yes or no? Yes or no? In response to the falling blood glucose level, your alpha cells of pancreas is going to release a messenger called as, yes or no? Yes or no? Okay? And in the stress of lowered glucose level, stress, it causes stress. Okay? Even increasing, hyperglycemia is also a stressful condition, lowered glucose level is also a stressful condition. Okay? In lowered glucose condition, in the condition such as like when your blood glucose level is low, epinephrine is also released. Okay? Glucagon specifically acts on liver, epinephrine acts on muscles. Sometimes epinephrine also acts on liver, but glucagon cannot act on muscles. Okay? Interestingly, these two of the chemical messengers are going to act on a transmembrane receptor, which is a 7 transmembrane helical structure, and the receptor is called as GPCR. If you want to note down the full form, it is called as G protein coupled receptor. What is it called? G protein coupled receptor. I am not going to dig in detail about the transporter because we are going to talk about in detail about GPCR in another topic called as glucose homeostasis or blood glucose regulation of blood glucose regulation. There will be a different topic. Okay, we are going to in detail we are going to see what GPCR does and all. In brief, I am going to say glucagon binds over here, causing the activation of an enzyme called as adenyl cyclase. Okay, imagine the adenyl cyclase is now active. The name of the enzyme is adenyl cyclase. Am I clear? The binding of glucagon activates adenyl cyclase, that becomes active. The adenyl cyclase now, after activation, converts concentration of ATP into cyclic AMP. Cyclic AMP. And cyclic AMP is called as the secondary messenger. Okay, it is the messenger provided by specific tissues on specific condition. It cannot enter inside the cell, so as a result it poses its action provided by another messenger that is called as secondary messenger. Am I right? Okay? As a result of that, the concentration of cyclic AMP increases inside the cytoplasm. Is that clear? The increased concentration of cyclic AMP causes the activation of a component called as cyclic AMP dependent protein kinase A. If you remember the name, I have told you in short pKa. जब मैं glycolysis का regulation बता रहा था, I have used this term pKa, protein kinase A. That is cyclic AMP dependent. So the complete name of this component is cyclic AMP dependent protein kinase A. Okay, that becomes activated into active pKa. Cyclic dependent protein kinase A. It is kinase, so what is the role it is going to play? It is a kinase, so what is the role it is going to play? It is going to transfer phosphate group. If we talk about transferring a phosphate group, there has to be a donor because it is kinase. So the donor has to be ATP, yes or no? It can be GTP, it can be ATP. Yes or no? So finally, if we are going to say that protein kinase A is going to transfer a phosphate group provided over a substrate, we call that process as phosphorylation, addition of phosphate group. Yes or no? So when we add a phosphate molecule as a phosphate group over a substrate, we call that component as to be phosphorylated state. Yes or no? Yes or no? So pKa plays an important role in conversion of, in activation of a component called as phosphorylase kinase B. ध्यान से देखिएगा. The enzyme name is phosphorylase kinase B. Upon the activation of pKa, what is pKa is going to do? It is going to add a phosphate group. Yes or no? Phosphate group is added. See? As a result, we can say that phosphorylase, short में लिख रहा हूँ, kinase is going to get activated in its state, and that activated state is called as phosphorylase kinase A. You can see a phosphate group over there. Is that easy to understand? Is that easy to understand? Glucagon got released responsible for, due to lowered blood glucose level. As a result of that, it binds to certain receptor that is called as GPCR. We also call that receptor as beta adrenergic receptor if it is in muscle. Epinephrine binds to beta adrenergic receptor that is same as that of GPCR structurally. 7 transmembrane helical structure, same, no difference. Beta adrenergic receptor or the GPCR activates an enzyme called as adenyl cyclase, which leads to the formation of increased concentration of cyclic AMP. Cyclic AMP is a secondary messenger that activates an enzyme called as pKa. Cyclic AMP dependent protein kinase A. Yes or no? Because it is a kinase, it is responsible for the transfer of phosphate group. As a result, it transfers a phosphate group by hydrolyzing ATP into an enzyme called as phosphorylase kinase B. Upon addition of phosphate group, the phosphorylase kinase B is converted into phosphorylase kinase A, which becomes activated. Yes or no? Clear? Have you seen this enzyme in any of the pathways of glycogenolysis or glycogenesis? Have you seen? Was that enzyme anywhere there in the process of glycogenesis or glycogenolysis? No, it was not there. Okay? The activation of this enzyme, the activation of this enzyme, the phosphorylated form, is later going to activate, मैं उल्टा लिख रहा हूं, इधर लिख रहा हूं, ठीक है, is going to activate an enzyme called as glycogen phosphorylase. If it is inactive, the state is called as B form. Remember, whenever I write small b, it confirms the inactive state of that enzyme. ठीक है? Upon activation of phosphorylase kinase A, again it is kinase. What it is going to do? Transfer a phosphate group. The donor should be ATP. Upon whom it is going to transfer? It is going to transfer a phosphate group over glycogen phosphorylase B. Yes or no? Yes or no? Karo. ATP will be hydrolyzed to form ADP, and as a result, glycogen phosphorylase is converted to its state called as A. That is active state. Add a phosphate group. Yes or no? So this is how glycogen phosphorylase becomes activated. And if glycogen phosphorylase is activated, what you are going to see? Glycogen will be broken down into glucose 1-phosphate or glucose 6-phosphate, let it be. So glycogen degradation is activated, provided by conversion of glucose into glucose 6-phosphate. Yes or no? Yes or no? So we can say that, sir, this is done. So in the presence of low blood glucose level, in the presence of glucagon, in the presence of stress, or in the presence of epinephrine, glycogenolysis gets activated. Breakdown of glycogen gets activated. Yes or no? Fine. What should be the reciprocal of this? If glycogenolysis, breakdown of glycogen is getting activated, what should be the reciprocal of that? Glycogen synthase has to be stopped. Yes or no? Yes or no? Fine. How? See, I am using green pen for glycogen synthase process. Okay, I am going to show you that how glycogen synthase get blocked, so glycogenolysis gets activated. Vice versa of it. Okay? pKa. अब एक बात सोच के देखो, if this component, if this enzyme is getting activated, protein kinase A, is it specific to that it is only going to add a kinase group to, a phosphate group to this enzyme only? No. This pKa is responsible to add phosphate group to all of the enzymes present in the cytoplasm at that moment of time. Everyone will get phosphorylated. Everything, whatever enzyme is present over there, inside at that moment of time in the cytoplasm, pKa will phosphorylate. Fine. So same like that, in this condition, if this is a cell, this is a cytoplasm, this pKa will also phosphorylate glycogen synthase. Yes or no? Are yes or no? Because glycogen synthase is present in the cytoplasm at that moment of time. So it will phosphorylate. Yes or no? Are yes or no? इसका काम है सेल के अंदर जितने भी एंजाइम्स है सबको फॉस्फोराइलेट कर दो, सबके माथे में एक फॉस्फेट ग्रुप ऐड कर दो. वो आंख बंद के कर रहा है, वो देख भी नहीं रहा है कि कौन सा एंजाइम है, क्या है, नहीं है. मैं एक्टिवेट हो गया मतलब मैं सबको फॉस्फेट ग्रुप ऐड करता जाऊंगा. वही कर रहा है. यहां पे किया तो ये बेचारा एक्टिव हो गया. ठीक है. ग्लाइकोजन सिंथेस पे करा, ग्लाइकोजन सिंथेस पे फॉस्फेट ग्रुप ऐड किया, ग्लाइकोजन सिंथेस बेचारा बन गया इनएक्टिव. ये एक्टिव था. You getting my point? So now what is the scenario? What is the scenario? What is the picture you are getting? The picture is in the presence of fasting, when your blood glucose level is low, that causes the release of glucagon, that causes the activation of pKa, increases the concentration of cyclic MP, causes the activation of pKa. The activated pKa activates by adding a phosphate group to glycogen phosphorylase, and glycogen phosphorylase becomes active, On the other hand, the pK also adds a phosphate group to glycogen synthase, making glycogen synthase inactive. So what is the scenario? The scenario is when your glucose concentration is low, glycogenolysis activates, glycogen synthesis drops. This is how the thing is going to happen. Yes or no? Yes or no? Am I clear to all of you? Is that point clear? Very important question. They will ask you in direct question they will ask you. Directly they will ask you this question regarding explain the reciprocal regulation of glycogen metabolism. Simple. Ekdam direct. Am I clear to all of you? Let us change the scenario. Okay. The scenario now we are going to look for. After surviving this fasting state, we now had something. We had now some food. Yes or no? We had something. Fine. So we are now into well-feeded state. We had something. When we had something, so as a result, when we had our food, the glucose concentration is going to increase. Yes or no? Okay? That will cause the release of insulin. Fine. Chalo. That will cause the release of insulin. Now insulin, just like GPCR, but it will bind to another receptor, surface level receptor that is present in, it will bind to a receptor that is called as tyrosine kinase receptor. What it is called as? ठीक है? Yes or no? Tyrosine kinase receptor to which the insulin is going to bind, provided to that it is going to activate at a later point an enzyme called as phosphatase, or it is also called as protein phosphatase 1. मैं एक जगह पे लिख रहा हूं, कंफ्यूज मत करना, ठीक है? See, so when we had food, blood glucose level increased, causing the release of insulin, which attaches to another surface receptor called as tyrosine kinase receptor. Fine. That activates the tyrosine kinase receptor, increase the activation of another cytoplasmic enzyme called as protein phosphatase 1. Just like glucagon was responsible for activation of pKa, fine, the insulin is responsible for the activation of cytoplasmic protein phosphatase 1. नाम suggest क्या कर रहा है? Kinases transfer. Phosphatase removes what? The phosphate group in the form of inorganic phosphate. In the form of simple. So now, as I told you, just the scenario change हुआ, function अभी तक change नहीं हुआ, it is still same. What I told you, that the pKa is going to add phosphate group to all the enzymes present inside the cytoplasm. Yes or no? I told you na. Same like that. Now the situation is shifted. Situation is we had food. As a result, insulin got released, causing the activation of protein phosphatase 1. Yes or no? So in this scenario, in this case, what protein phosphatase 1 is going to do is, it is going to remove phosphate group from all the enzymes present inside the cytoplasm. Is my point clear? Is my point clear to all of you? Fine. Now see the scenario. In the presence of protein phosphatase, fine. Blue se likh raha hu. Protein phosphate group will be removed from glycogen synthase. Yes or no? Are yes or no? Making it, so glycogen synthase is active. We had food, no? So glucose should be used to store back our glycogen in the liver. Yes or no? Got the scenario where that link coming from? This is how insulin or hormone plays a very important role in covalent modification. This entire process, what you are seeing, we call them as covalent modification. Okay? So the phosphatase will remove the phosphate group, leading to the activation of glycogen synthase. If a glycogen synthase is activated, glucose 6-phosphate will start converting into glycogen. Start. But we cannot start this without inactivating this. So first we have to inactivate it. Otherwise, imagine karo कि इधर से glycogen synthase working and on the other side glycogenolysis is working. What is the net effect? Zero. What is the net gain? Zero. So for that reason, before you initiate glycogen synthesis or start producing glycogen, synthesizing glycogen, you are supposed to stop glycogenolysis. How? See, protein phosphatase, what it will do? It will cause the removal of phosphate group from phosphorylase kinase A. Yes or no? It will remove the phosphate group, making it inactive. When phosphorylase kinase A is inactivated to this, it cannot add further phosphate group to glycogen phosphorylase. As a result, the phosphate group from glycogen phosphorylase will be removed, making it inactive. When glycogen phosphorylase B is inactive, when the glycogen phosphorylase is inactivated, provided by removal of phosphate group from glycogen phosphorylase A, the glycogenolysis process halts, stops. Am I clear? लेकिन यहाँ पे एक point आता है. What was my statement just before I started with glycogenolysis? Now, my statement was that before we should activate glycogen synthase by removing the phosphate group, first of all, we must stop it. Stop this phosphorylation. Stop this enzyme cascade reaction. And this enzyme cascade reaction is responsible, whole total responsible of increased concentration of cyclic MP. Are you getting my point? अगर cyclic MP का concentration नहीं बढ़ता, तो क्या ये एक्टिवेट होता? कहाँ गया? क्या ये पीके एक्टिव होता? अगर पीके एक्टिव नहीं होता तो क्या ये फॉस्फोराइलेशन रिएक्शंस होते? एडिशन ऑफ फॉस्फेट ग्रुप होता? नहीं होता. So the first thing that insulin is going to do in the form of certain secondary messengers is it will activate an enzyme called as phosphodiesterase, which will cleave down the cyclic AMP into AMP. As a result, the first concentration of cyclic AMP will start falling down. So because the concentration of secondary messenger is started falling down, the activation of protein kinase A will become the activation of protein kinase A becomes halted, or the protein kinase A will become inactivated. When protein kinase A is inactivated, it cannot further add phosphate group to phosphorylase kinase. It becomes inactive. As a result, this also becomes inactive. Are you getting my point? Is the scenario clear? If you cannot understand this in this way, you go through book, it will be more tough. I have explained in a very, very simpler way. If any part of process, if any of the part of process, if you are unable to understand, please raise your hand. Feel free. If glucose condition is low, yes, then what are you asking? Glycogen level is?
So that important part, not health conditions, including the cycle.
Acha, you are going to answer me this. Okay. Ek scenario, you are not, what is the biochemistry is a subject where you require a better approach, because biochemistry you can't see, you have to imagine things. Yes or no? In this scenario, just what she is asking, like in the presence of low glucose level, what is the status of glycogen, whether it is low or high, in the presence of glucagon?
So it will be active or inactive something.
Now imagine, you didn't have food, your blood glucose level is low, your brain cells need energy in the form of glucose. Yes or no? So your glucagon before this scenario, when you didn't had food, तो कुछ दिन पहले तो खाए थे ना, तो when you had food, your all glucose, excess glucose was stored in the liver in the form of glycogen. Yes or no? Huh? Suddenly you stopped having food. You are in fasting state. In that scenario, your brain requires glucose. Your RBC is totally dependent on glucose. As a result of that, what will happen? The glycogen that was stored from beginning time period, it will get broken down. Is that clear? Fine. Okay. Is my explanation clear to all of you? Is that what you are expecting? So in other ways, we can say that when the condition is hypoglycemia or your lower blood glucose level, the glycogen will be broken down so that the glycogen can convert into glucose or glucose 6-phosphate. Okay? If this process is occurring in liver, the glycogen will be converted into glucose. The glucose will go out of the liver into the blood. But if this process of glycogenolysis is occurring inside skeletal muscle, the final product is glucose 6-phosphate because muscle cells lacks glucose 6-phosphatase. Am I clear? Now, after this, when glycogenolysis was over, you had something. The liver demanded that, okay, my glycogen store is khatam, I need to store it back. As a result of that, in the presence of insulin, glycogen degradation will stop and glycogen synthesis will start. So when you add food, again the glycogen store is fully packed, leaving your liver cell full. The tank is full, tank is full, provided for the next period of fasting, making your liver ready for the next period of fasting. Am I clear? What are you asking? Acha. Okay, got you. Phosphodiesterase is an enzyme that is activated by insulin. Did you got my point? This enzyme is activated by insulin. Fine. And phosphodiesterase is responsible for lowering the cyclic AMP concentration. How it does? It converts the cyclic AMP into 5' AMP, making the hydrolysis of cyclic AMP. As a result, till the moment this enzyme is activated, whatever cyclic AMP concentration was there inside the cytoplasm, provided by, that was made when glucagon bind to the GPCR, was cleaved, is going to get hydrolyzed into 5' AMP, making the fall in concentration of cyclic AMP. The cyclic AMP concentration will fall down, and this will take place in the presence of insulin. Okay? Now, if cyclic AMP concentration is going to fall down, what will happen? It is not going to activate this enzyme. It is not going to activate pKa. pKa will not get activated. Yes or no? So, if pKa is not going to get activated, this enzyme won't get activated. Yes or no? If this enzyme is not going to get activated, this enzyme will not get activated. What is this enzyme? Glycogen phosphorylase. Glycogen phosphorylase is an enzyme that is responsible for breakdown of glycogen. So when, in other term, you can say, in the presence of insulin, breakdown of glycogen will stop. In presence of insulin, formation of glycogen will start. Yes or no? Am I clear to all of you? In other way, I just want to let you know that there are very limited, you can count for the syllabus that you are going through, or you are going to be, you are studying the syllabus, whatever syllabus you are going to cover in biochemistry, there are limited enzymes which are activated when they are in phosphorylated state. Out of those 5, I can count you 5. Out of those, 2 enzymes are here. One more enzyme is there, that is in glycolysis regulation, fructose 2,6-bisphosphatase. Remember that bifunctional enzyme, one house but 2 roof. Remember that example? Remember, yes or no? 3 examples are already here. ठीक है? There are very limited number of enzymes which are active when a phosphate group is added upon them. Otherwise, the majority of the enzymes that are present in our body inside the cell are active when there is no phosphate group added to it. You can take the example of glycogen synthase. There are many other, almost all. The exceptions are only 5. Glycogen phosphorylase is active when a phosphate group is added. Phosphorylase kinase, fructose 2,6-bisphosphatase in glycolysis, and another that you are going to study in lipid metabolism, specifically lipoprotein metabolism, that is called as HSL, hormone sensitive lipase. 4 मैंने बोल दिए अभी, और एक है, आगे पढ़ोगे. ये 5 ही है. There are only 5 specific enzymes which are active when a phosphate group is added. Otherwise, all other enzymes are active when a phosphate group is not there, or they are active when they are in dephosphorylated state. Okay, remember this. This is what about covalent modification. Now you think about allosteric modification. Both of these enzymes can play a very important role provided to their activation inactivation in the presence of allosteric effectors. You are going to say me, I told you, you have to understand the things specifically. Now you imagine what should be the allosteric effectors of glycogen phosphorylase. Think about it and say me, answer me. Allosteric effectors. सोच के देखो. Now see, the process is conversion of glycogen into glucose 6-phosphate or conversion of glucose 6-phosphate into glycogen. There are 2 processes. If this is the process, we call it as glycogenolysis. Yes or no? मैं short में लिखूँ. Glycogenolysis. If this is the process, we call it as glycogenesis. Hormonal modification, covalent modification, regulation, I have told you. Now coming to the allosteric activator, because both of these enzymes are the key enzymes and are responsible for allosteric activators or inactivators. They can be regulated allosterically. ठीक है? You are going to say, see, glycogen will convert into glucose 6-phosphate. What do you think the scenario of the cell should be? Be fast. Whether the cell is in high state, whether the cell is in low energy state, what? Low energy state बोले तो decreased concentration of glucose 6-phosphate, yes or no? अरे yes or no? So can we say that increased concentration of glucose 6-phosphate inhibits it and decreased concentration of glucose 6-phosphate activates it? अरे yes or no? Can we say increased concentration of glucose 6-phosphate is going to inhibit it? Yes or no? Is my point clear? If your cell is having high glucose 6-phosphate, your cell have lot of glucose. Your cell is having lot of energy as well as lot of glucose. Enough. In that scenario, do we need further glucose to be produced? So why to break down the glycogen then? Rather, we can use the glucose 6-phosphate to form glycogen and stock it and store it. Yes or no? So just iska reverse kar do. If increased concentration of glucose 6-phosphate, it will act as a positive effector or not? Are yes or no? Increased concentration of glucose 6-phosphate gives an idea about that the cell is having lot of energy. No need of further going to glycolysis. Cell के पास already बहुत सारा ATP है. दोबारा और ATP बनाने का जरूरत नहीं है. Rather, we can serve, we can stock, we can store the form of energy in form of glycogen. It's an idea, it's an information the cell is giving out. You're getting my point? Fine. Just reverse it. So lowered glucose concentration will, glucose 6-phosphate concentration will cause? Lowered glucose 6-phosphate. Whatever glucose was there is now utilized. The cell is now lacking of glucose, lacking of energy. In that point of time, it will give a signal, it will inactivate glycogenesis, synthesis of glycogen, and will activate glycogen breakdown. Easy है. थोड़ा सा सोच के देखना. ठीक है? Now in terms of energy, if your cell is already having lot of ATP, high concentration of ATP, If your cell is having high concentration of ATP, whether it will activate glycogenesis, synthesis of glycogen, or it will inactivate. Good, very good, you are understanding. How many of you are going to say that yes sir, increased concentration of ATP is going to activate the process of synthesis of glycogen? Raise your hand. Who stands with me? High concentration, very good, you are slowly you are taking it up, very good. High concentration of energy. Already cell ke andar your body is having lot of energy. Do you want more energy? In that way, so to reduce the concentration of energy in your body, what you will do? You will do exercises. Yes or no? Same thing is happening. Your body is having lot of ATP. No more need of glycolysis. No more need of production of ATP. Rather, save it. How you are going to save? By converting the glucose 6-phosphate into glycogen. Simple. Am I clear? Fine. High concentration of AMP is going to act as a inhibitor or activator. How many of you say? How many of you say? High concentration of AMP, what does signal says? That the cell is having lowered energy. Yes or no? Low state of energy. Cell के अंदर energy कम है. Cell को energy बनाने की जरूरत है. Cell is in need to provide or to produce energy. In that scenario, this is a synthetic process. This is an anabolic process. Anabolic process requires ATP. Already cell is lacking of energy. And in that meantime, if you keep going for this reaction, the cell will require more energy. And the cell is already lacking energy. Fine. So in this scenario, what will happen? That the increased concentration of AMP will bind as a negative allosteric effector, will cause the inactivation of this process, bole the synthetic process, and it will activate the glycogenolysis. You are getting my point? Simple. Simple as it is. Is that easy for you guys? Can anyone? अच्छा, one more thing I have forgotten. देखो यहाँ पे correlation है, देखो कितना मजेदार correlation है. Increased concentration of AMP for glycolysis, if you remember, is an allosteric activator of an enzyme called as PFK1. Yes or no? Increased concentration of allosteric, AMP. या फिर हमने इसको ऐसे लिखा था. This can be also written in this way: decrease ATP by AMP ratio. बोले तो the concentration of ATP is less and the concentration of AMP is high, explaining about the state of the cell as low energy state. Fine? Is that clear? This activates PFK1. PFK1 is an enzyme responsible for the rate limiting step of glycolysis. Yes or no? Yes or no? So if PFK1 is activated, what you expect? That the glucose will convert into pyruvate or will get oxidized. Yes or no? Making the production of ATP. Yes or no? Yes or no? You're getting the correlation. Glycogen breakdown is getting activated by AMP, increased concentration of AMP, lowered energy state. A result is glucose 6-phosphate. And in the meantime, so that the cell will produce energy in the form of ATP, will reverse this condition, will reverse the condition. For that, your cell requires glycolysis to be activated. You are getting my point? यहां से ग्लूकोज 6-फॉस्फेट बना. अब इस ग्लूकोज 6-फॉस्फेट को अगर सेल यूटिलाइज ही ना कर पाए तो क्या अचार डालेगा इसका? You are supposed to utilize it. So for utilizing glucose 6-phosphate for the production of energy, you require glycolysis. See, glycolysis is getting activated. So glycolysis in meantime, साथ में साथ parallelly gets activated, so that the glucose 6-phosphate that is formed goes into glycolysis and provides ATP. As a result, the concentration of ATP inside the cell is going to increase. Am I clear? Last point that you are going to explain me, saying what is the role what you understand, if there is increased concentration of calcium, it is going to activate glycogen synthesis or it is going to activate glycogen breakdown. Say me. Anyone? What is the role of calcium? It is going to activate glycogen phosphorylase or breakdown of glycogenolysis, or it should activate glycogen synthesis. Calcium, think about skeletal muscle. Sarcoplasmic reticulum releases calcium when you are going for excessive contraction. Yes or no? Fine. When your muscles are extremely contracting, you are going for certain exercise, you are performing certain exercise, your skeletal muscles require ATP. Yes or no? Yes or no? So that means if the cell is in requirement of ATP, what do you think? The glycogen should be synthesized or it should be broken down? It should be broken down. All of you agree? Yes. So increased concentration of calcium, what is going to do? Is going to activate glycogenolysis. Yes or no? Fine. This calcium will specifically bind to the calmodulin area of this enzyme. Okay, 4 calciums are going to bind to a specific calmodulin complex, forming calcium calmodulin complex that will activate glycogen phosphorylase without covalent modification also. Am I clear? There is certain diseases specifically for glycogen synthase metabolism glycogenolysis metabolism that we call as glycogen storage disorder. Very commonly asked question for your first professional examination, as well as very commonly asked question for the exit exam that you are going to write, or maybe I don't know, but for NEET examination, for NEET preparation, glycogen storage disorder, GSD, is very important, out of which the most important is von Gert's disease. Okay, von Gert's disease. That is due to the deficiency of glucose 6-phosphate. I have told you already, phosphatase. I have already told you specifically in the last class, but now I just want to clarify you one thing. You must, in the book, if you see Vasudevan, they have written 2-3 points over von Gert's disease. That is, due to the deficiency of glucose 6-phosphate, what happens exactly is, it causes fasting hypoglycemia that doesn't respond to adrenaline injections. What does it mean? What does it mean? Glucose 6-phosphatase is an enzyme responsible for breakdown of glucose 6-phosphate to glucose. Yes or no? It is an hepatic enzyme. Yes or no? This enzyme is also responsible, it plays a very important role in gluconeogenesis. Yes or no? So if this enzyme is absent, specifically what will happen? That glucose 6-phosphate will get trapped inside the liver. It won't go out of the liver. If it is not going to go out of the liver, your liver is responsible to regulate the blood glucose level when you are in fasting state, to increase the blood glucose level when you are fasting state. So if glucose 6-phosphate is not going out, glucose 6-phosphate is rather not converting into glucose. In the meantime, glucose is not reaching the blood, so the blood glucose level falls down. This is called fasting hypoglycemia. This is called fasting hypoglycemia. And provided to that, if you gave adrenaline shots, this pathway is not going to affect anything. If you give adrenaline, it attaches to the beta receptor, causes the formation of cyclic AMP, everything, this all thing will happen, leading to the breakdown of glycogen into glucose 6-phosphate. Everything will happen. But the liver glycogen will convert into glucose 6-phosphate only. Glucose 6-phosphate cannot move out of the cell. As a result, it will not contribute to increase the glucose concentration in the blood. So, even if you are giving a shot of adrenaline, it will not respond to the fasting hypoglycemia. Okay. There are certain conditions that they explain with them that comes up with this von Gert's disease, that is hyperuricemia. Because there is excessive glucose 6-phosphate concentration in the cell, it is not going to get utilized in that state of time. It will move to another pathway called as HMP shunt. This HMP shunt is responsible for production of ribose 5-phosphates. and these ribose 5 phosphates are responsible for the production of purines and pyrimidines, nucleotides. Okay, so excessive purine will get formed. So if excessive purine that is not getting utilized, the body will start catabolizing it, will start breaking down it. So the end product of purine catabolism is nothing but uric acid, not urea. Uric acid is the end product of purine catabolism. Okay, that will lead to increased concentration of uric acid in your blood, causing hyperuricemia. That will lead to cause of gouty pains and all. Okay, there is one more condition, says that it also causes lactic acidosis. Lactic acidosis is caused because lactic acid and uric acid competes with each other to get released out of your body. So as there is already a higher concentration of uric acid in your blood, the lactic acid will not be allowed to go out of your body through the urine. There is a high competition. As a result, the lactic acid stays inside your blood, leading to lactic acidosis. ठीक है, वाइवा में ये क्वेश्चंस बहुत पूछते हैं, why lactic acidosis is caused and all sort of things. Okay, thank you, and if any doubt, please feel free to ask.